So I'm Maaly Nassar. I'm a senior data scientist at Elsevier. And then today I'm going to talk very fast on the streamlining of drug development with conversational AI-powered knowledge graph. So basically, I will start by going directly through the drug development process, and highlighting the critical phases to its failure or success. And then I will show you how we develop this knowledge graph and augment or power it with AI, and why we are doing this. And then I will go through the use cases of the knowledge graph with our pharma customers. But before jumping into my talk, I just want to tell you who we are. So we are SciBite. We are a company from Elsevier, and we basically offer a cocktail of bespoke solutions with our software, with our data, and with our data science services. We license three main softwares, which is Sentry, the ontology management system, Termite, the name entity recognition, and SciBite Search, which basically integrate our curated ontology and name entity recognition to retrieve semantically relevant articles. We also license ontologies curated, which align with the FAIR principles and machine learning vocabularies. And on the top of that, we offer data science services like the AI-driven services and analytics, and mainly through knowledge graph. So why do we need knowledge graph in the drug development? So basically, we want them to correctly identify the causal link between drug, target, and disease. Why is that? So there is a study that has been done by FDA, and this study basically said that 63% of the approved drug are supported by causal links between their targets and the disease. But despite this, the clinical trial exhibit a 90% failure rate. And this is due to the poor prediction of underlying cellular machinery and unmanageable toxicity. And that's why we need to clinically translating and validating drug target cellular machinery within extensive scientific literature. So, what I'm going to say, I feel that I'm hearing the echo of my voice. So do you hear me clearly? Hello? Do you hear me?
We hear you just fine. I'm turning off the audio on our end, and we have maybe 5 minutes more in the talk.
Okay, so basically, maybe I should skip this one. So to develop the knowledge graph, we basically index all the data sets. You can see a lot of BMC and PubMed and so on. And we index them. We extract the sentences and the entities from the text using the SciBite search. And then we, on the top of that, classify the relationship with embedding models and fine-tuned models to answer the questions of the customers. And we use also this embedding models to score the relationship. And we use the score for the graph analysis to pinpoint key targets. We have compared different embedding models, like 14 embedding models, and you can see the top 5 embedding models are coming basically from OpenAI and Flag embeddings. I used BioAsk dataset for the evaluation to see exactly which embedding model is performing better by comparing the results retrieved by each embedding model and the results associated with the BioAsk dataset. And you can see here it is 66%, which is not really good, but it is because I was comparing the same number of results retrieved from the embedding models and also the same number of results coming from the BioAsk. But if I just see the overlap between the top 50 and the BioAsk PubMed ID, I get 80%. Based on that, I have used Flag embeddings with many customers. And the first use case I had was a preclinical target identification, where the customer basically asked for genes upregulated and downregulated in specific disease. So we built this user interface on the top of the knowledge graph, where the customer actually asks a question, and then he gets all the results from the literature with the co-occurring entities and the summary for the answer of the question. So here, the whole interface starts by the question being translated into embedding, indexed into Neo4j vector search. Semantic similarity metric is calculated between the question and the context and the co-occurring entities are retrieved and summarized. And I've used the same interface actually to classify and score the relationship. So here, instead of using the question, I use the class, whether it is upregulation and downregulation, to classify the relationship between all the entities in the graph. And basically, it is like the score is a semantic similarity between the class and the context. And once I classify and score all the relationship, I can use the score for the graph analysis to prioritize targets or drugs based on direct and indirect relationships. Regulation is a very complex topic. So to decipher the regulatory effect between two entities, it is not that straightforward when you get it with embedding models. And for that reason, what I've done here is basically I've used large language models to generate output or to try to identify the relationship between two entities. And I use the judge model to assess these outputs and decide which output is the right one. And then I've used this judge data set to train or fine tune other models. And you can see that the precision is between 80 to 95%. I've tried also to compare the large language model as a judge with the human judge. And you can see that the human judge really aligned with the large language model. This is another use case for microbiome repurposing, where we have used the graph analysis with the therapeutic score to pinpoint the therapeutic metabolites and microbiome bacteria for specific diseases. The validation of this, I usually do it with, I validate with the genomic sequence analysis. For the microbiome, I've used Magnify. Magnify usually do metagenomic analysis and kind of summarize all the bacteria with their counts in the samples. So I basically validated the bacteria in the samples with the bacteria coming from the graph, and there was large overlap between them. Most of the validation is coming from the customers, where they provide testimonials about our work and when they validate their experimental results. The other use case is basically matching patients to clinical trial. It is the same scenario, but here, instead of using context, we use the eligibility criteria of patients and clinical trials. And here we first started by matching the entities of the patient eligibility criteria with the trial eligibility criteria. This didn't quite work because we need to capture the semantics. We used the embedding model to capture the semantics of the eligibility criteria. This also didn't accurately work in matching patients to clinical trial. So on the top of that, we have fine-tuned large language models or use off-shelf large language models like GPT-4 to assess the criteria between patients and clinical trials and to see exactly if they match or not, eligible or not, and why. And this actually produced better results. And this is very naive analysis of the count of the trial.
Sorry to interrupt, but can you wrap up, please?
Yeah, I've just... So this is basically the drug toxicity. The same procedure happened. We use also the question answering and also to classify the relationship and scoring for graph analysis. We validate against the database. And in summary, so I just go very quickly over the potential of large AI-powered graph in drug development. We still are looking for benchmarking data sets for increasing the precision. And you can see that our collaboration of customers have been the driving force for such development. Yeah, thank you for listening.